Chapter 1 is an intro to biology chapter where we'll study biology and what it's about. We'll study the themes in biology, and we'll study evolution as the foundation of biology, as well as the scientific process. This is a picture of the author of your textbook, Neil Campbell. Biology is the study of life. Bio means life, and ology means to study. So that's where that word comes from. Living things have certain characteristics. So we call those the characteristics of life. Number one, living things are made up of cells, at least one. So it can be a unicellular organism, for example, a bacteria, or multicellular organisms like frogs, like you. All living things must be able to reproduce, so either asexually or sexually. All organisms grow and develop. Growth specifically refers to an organism getting larger in size. And so, for example, first of all, a cell does grow, but then one cell divides to make two cells divides to make four cells, 4 divides to make 8 to 16, and so on. And so as an overall additive factor of that, the organism as a whole grows. Whereas development refers to an organism becoming more different, right? Organisms change over time. You're very different today than you were when you were 5, than you were when you were 2, than you were when you were an infant. And you will continue to change and develop throughout your lifetime. Here's another example showing development. You can see that this frog is developing from an egg stage all the way through to tadpole and then to an adult organism. All organisms have metabolism and utilize energy. So metabolism is kind of a general term. It can include either building something up or breaking something down. So a good example of metabolism would be something like cellular respiration, where your cells are breaking down sugars into usable packets of energy. Or digestion, where you consume food and then your body breaks it down into smaller pieces so it can absorb nutrients and further break them down. And then energy utilization would be like actually using that energy that was created in cellular respiration, right? So you need energy to do a lot of things. You need energy to write your notes. You need energy to have your cells divide. You need energy on multiple levels. And so this is what that's talking about. Living things respond to environment. So a good example of that is shown here, right? So plants have this unique capability to respond to the sun and grow upwards, right? So you can see that this plant is growing up. If you've ever seen trees that grow on like the side of the mountain, they still grow, pretty high up. They don't grow out, even though the mountain has a very steep side. Whereas the roots of the plants typically grow down into the soil because they need to access the water. So they're responding to their environment. If you've ever seen sunflowers in like a slow motion video, they actually kind of turn their leaves and flowers towards the sun throughout the day because the leaves are able to absorb large amounts of sunlight, and so again they're responding to their environment. Homeostasis, this refers to an internal balance to your external environment, right? So there are lots of different types of homeostatic mechanisms in different organisms, but a really common one, a classic example to use, is how your body is actually able to maintain a stable temperature. Like your internal body temperature stays the same no matter how hot or cold it is outside. Your body has certain mechanisms in place to maintain homeostasis. So when you're hot, for example, your body sweats to cool you down, and when you are cold, your body shivers to try and warm you back up. It's trying to maintain homeostasis. Evolutionary adaptations refer to changes in a population or group of organisms over a period of time. These refer to specifically advantages that are passed on from generation to generation. Advantages that increase survival and capabilities of reproducing for a specific organism. So you could probably think of a lot of adaptations. So it's basically anything that would be helpful to the organism. So a good example would be something like a cactus has spines. So they've learned to, or they have the ability to develop spines, which is a great adaptation because it protects them against predators. Cacti actually don't have very many predators. They also are able to conserve large amounts of water, which is why they're able to survive in very dry areas like deserts. An example of an evolutionary adaptation in an animal could be something like the counter shading that sharks have. Their underside is typically much lighter than their dorsal side, the backside, and this helps them to be able to sneak up on their prey. So it's like kind of similar like when you're in the ocean or in a swimming pool even, and you look up, it's very bright, right? So when a fish looks up at a shark, their belly is light, and so that helps them to sneak up. Or if they're traveling under the fish, right, when you look down in the ocean's very dark. So the top side of the shark being dark would be a great advantage because it allows it to sneak up on the fish. So next we'll talk about themes in the study of life. Your textbook does a really good job of organizing this information as well. So it's a good resource if you want extra examples or support throughout this chapter. Number 1, the first theme is life is organized on many structural levels. Okay, so from small to big, each different level is very organized. So I'm going to explain each level. Atoms are the smallest level. Atoms are the basic unit of matter. They are not considered alive, but they make up everything, right? So atoms make up molecules. Specifically in this year, we'll really focus on biological molecules because this is a biology class. So atoms make up biological molecules for this class, but they really make up all molecules. So examples of a biological molecule could be something like a carbohydrate or a protein or DNA. Molecules make up organelles. Organelles are small kind of compartmentalized structures that have capabilities of performing pretty specific functions. So good examples of organelles would include something like the mitochondria, or the Golgi apparatus, or the nucleus, something like that. Subcellular means it's inside the cell. It's below the cellular level. So it's still smaller than the cell because organelles are found inside of cells. And so cells is the next level of organization. Cells is the first level that is considered alive. So nothing below the cellular level is alive because it's not capable of performing life's functions. Cells can perform life's functions, so cells are considered the smallest living unit. Cells then make up tissues. Tissues are a collection of the same type of cells. Tissues make up organs. So here's an example of an organ. So we have the heart right here. So you can see we have cells, and then there's all these different types of cells. This is a tissue. this is the tissue, this is a different tissue type, a different tissue type. But then all these different tissue types have to come together and function together to make an organ do an overall function. Tissues can't do alone. Each individual tissue type cannot perform the function alone that your heart can actually perform. And then the next level of organization is organ systems. So your body has a lot of different organ systems. So here you can see we have the circulatory and respiratory systems, right? So you can see how the blood is flowing through the circulatory system, and the heart is just one portion of that. But in an organ system, you have multiple organs working together, right? So we have our lungs here, we have our kidneys here, all of this works together with the heart to be one functioning body system or organ system. And then organ systems come together to make up the organism, complex organism that can perform a lot of different functions because of all these different organ systems working together. So it's no longer just one system. It's not just the circulatory system, but also the respiratory system, the digestive system, the endocrine system, the reproductive system, all of them. There are more levels beyond the individual organism, right? It doesn't stop just there. A group of organisms that all belong to the same species and live together in the same area is called a population. So it has to be same species, and they have to live in the same general area. If they live in different areas, they may still be considered the same species, but they're not members of the same population. So a good example of this would be something like there's lions that live in northern Africa, and there's lions that live in southern Africa, and they are the same species, but they don't live together. They live completely different sides of the continent, and therefore they're two completely different populations. The community is the next level. A community are groups of populations that live together in the same general area. So now we're not just talking about one species, right? It's multiple populations of different species. So maybe the lions and the zebras and the antelope and the grasses and the trees and the elephants and the bacteria that all live together in the African grasslands or in the savanna. That's a good example of that. Communities make up ecosystems. An ecosystem is essentially the living organisms in the community, so we call that biotic factors, and all the non-living factors or abiotic factors that work together with the biotic factors. So again, an ecosystem includes all the organisms in the community plus everything non-living that helps it function, like the rocks, the water, the air, things like that. And then we have biomes. Biomes are a very specific geographic area where certain ecosystems are found. And it can be many ecosystems. So a good example would be something like in an aquatic biome, it might have ecosystems of coral reefs and kelp forests, right? So ecosystems can, collectively in the same area be considered what we would call a biome. And then next level is the biosphere. The biosphere includes the entire Earth, essentially, both above and below Earth's surface. The next theme is emergent properties. So as we move up the biological hierarchy, so the different levels of organization, new properties emerge that wouldn't be possible if you had stopped at the last level, right? Because it becomes more complicated and more things are able to work together and create a more complex level at the next level. So the example shown here is a cardiac muscle cell, which is capable of contracting and performing other important functions, but it can't really do a lot on its own, just one cell. when it comes together as a tissue, they can contract together, which is something it couldn't do in the prior level. Then we have different tissue types, including this specific muscle tissue, plus other tissue types coming together to make up this organ, the heart, right? And so the organ is much more complicated. It's able to pump blood to both the lungs as well as out to the body. And so once that pumping has taken place, the vascular system here, the cardiovascular system, works together with the heart to be able to take all that blood, which carries oxygen, to the body, to different parts of the body, and then return that deoxygenated blood to the lungs. And then here we have an organism, which obviously could not function without all of these things, and with other systems that also go through these different levels of biological hierarchy. Even more properties will emerge when you consider them collectively as this complex organism. Oops, going back. Emergent properties lead to the characteristics of life. So it leads to things such as being able to actually reproduce to make more cells. It leads to the ability of organisms to grow and develop, so become more different over time. It allows organisms to be able to metabolize food and then use that food for energy and respond to the environment, because all of these different things work together, and previous levels cannot do what the next level could do. So it just continues to become more complex as you move up the levels. The third theme is that cells are the basic units of structure and function. So here we have pictures of cells. You can see that we have a eukaryotic cell here. We have a prokaryotic cell here. And then here's another picture, or like a model, of a eukaryotic cell as well. Cells are incredibly complex, but they are the smallest unit that is considered alive. The cell theory has 3 parts. Number 1, the cell is the lowest level of structure capable of performing all activities of life. So again, nothing smaller than the cell can perform all of life's functions on its own. A cell is fully capable of doing that. So when you consider the next level down, which is an organelle, like a mitochondria or a nucleus, shown here, these things have to work together to make one functioning cell. Number 2, all organisms are composed of cells. All living things are made up of cells, at least one. Bacteria are made up of one cell, but many eukaryotes are made up of more than one cell. Number 3, cells come from pre-existing cells. Life comes from life. Cells must divide to make more cells. That's what that means. And then down here, it talks about the definition of a theory. It says a theory is a well-substantiated, sorry, it's being blocked right now. Whoops. Going back. A well-substantiated explanation of some aspect of the natural world based on a body of facts that have been repeatedly confirmed through observation and experiment. So if you were to look up the word theory in the dictionary, you may get 2 definitions. One's going to say something like a mere conjecture or guess, but then the other one's going to say in science, right? So the cell theory is a scientific theory, and what that means is there's overwhelming amounts of evidence, multiple lines of evidence. There's so much evidence that it's unlikely that the theory will be disproven. How does this paramecium, this single-celled organism, perform the functions of life. So if you wrote your note takers, printed them, I would recommend on this slide in particular, like you put a big star and you write IB. Because if you plan to take IB year 1 and year 2 and then test in IB Science or in IB Bio, then you're going to need to remember this example. So that's why it's in here. It came straight from the IB textbook and the IB curriculum. So it's not like super important that you like understand the paramecium right now, but it does show you great examples, and I wanted to introduce it to you so that you're able to learn about it where it actually fits in the content. So we're looking at a paramecium, which is a single-celled protist. This particular organism lives in water. I want you to notice this it's called a contractile vacuole, but basically you can see that there's an animation here which shows this particular organelle moving, right? It's in motion. It's like almost like a pump, like expelling excess water. So I'm going to talk about that specific organelle in just a few minutes. Okay, so this just kind of goes through different functions that it's able to actually carry out that we've talked a little bit about. So let's start here at nutrition. Paramecium are capable of taking in food molecules, so things that are smaller than it, and therefore once the food comes in, it's able to then metabolize that food, break it down into energy. It uses or does a lot of its functions for many metabolic processes right in its cytoplasm. It's such a small organism. And then after it's absorbed the nutrients that it can, then waste will also be expelled almost in an opposite fashion of how it came in. So, through the cell membrane. And then homeostasis. One of the things that's really neat about paramecium is they constantly live in water. So they have to be able to maintain a balance. If they had too much water go inside the paramecium that couldn't come out, then the paramecium would just swell up with water, and eventually it could burst and die because the cell membrane that surrounds it is very flexible. So it's kind of like blowing up a balloon and just continuing to blow it up until it pops. But instead, that doesn't happen because they have a special organelle that's called a contractile vacuole. And what it does is it pumps and expels the excess water. That way it's able to maintain the proper volume ratio that it needs to be able to live well in the water. And then response to environment. If you see these little kind of hair-like projections, they're kind of clear, kind of translucent, but they're all around the paramecium. These are called cilia, and they kind of wave around in response to changes in the environment. And so it can actually use this for mobility. So if it feels a change in the water, and that means that there might be food nearby, it can use the cilia to help move it towards the food and then help to also bring it inside the organism. And then it does reproduce. Paramecium have capabilities of performing both sexual and asexual reproduction. And then they also grow. So after they take in enough nutrients, they will actually grow larger in size until at some point they're too big. And then once they're too big, they don't quite function as well. And so at that point is when they would divide to make 2 more paramecium, typically through mitosis. This just shows you the life cycle. So again, it can undergo sexual reproduction, but it's also often asexual reproduction. The fourth theme, continuity of life, is based on heritable information in the form of DNA. So what that means is life continues because DNA can be passed on from generation to generation. The fifth theme, structure and function are correlated at all levels of biological organization. Form fits function. Structure leads to function. So that basically means that things have a certain form or a certain shape because that shape provides a very specific function. So this is true for biological levels, but it's also true for non-biology examples. So this bike example is pretty good. You could see that this bike has a much thinner frame, thinner tires. It's kind of sleeker, like you can see the seat and the handlebars are kind of at the same level. The handlebars sort of tilt down a little bit. This bike is meant for bicycling on the road, whereas this bike is different. It has much thicker tires, a much thicker frame, shocks. The handlebars kind of point up higher than the seat. This bike is meant for much more difficult terrain, like off-roading, rocks, things like that. Here are some biological examples. Here we have 3 different types of cells: skin cell, a nerve cell, a muscle cell. Your skin cells tend to be kind of flat, don't really have too much of a shape, just kind of more round. Their function is protection. This nerve cell has a very specific function that relates to its shape. It has to have longer areas which help to fire off signals that it can use to communicate to other cells, to tell other parts of your body to do something or for you to feel something. Whereas a muscle cell, they tend to be long and slender. They also tend to attach to bones and things like that, and they have capabilities of contracting, so they're used for movement. And then here shows you 2 organ systems. Here you have your skeletal system versus the digestive system. Your skeletal system is used mostly for protection, stability, support, whereas your digestive system is used to break down foods into smaller parts, absorb nutrients, eliminate waste. Here shows you an organism, a plant. You can see that there's this flower which has a very specific shape. That's because it attracts a specific pollinator. It has this flat surface right here, which is really good for attracting bees, whereas these flowers, they have like a more slender sort of pollen tube that is kind of hard to see in this picture, but they really wouldn't be able to attract bees because bees can't get in there and access the food that they would want to have. So instead, this is better for like a hummingbird, which has like a long, slender beak it can use to get deep within there. Number 6, organisms are open systems that interact continuously with their environment, which essentially means that organisms aren't vacuum sealed, right? They are constantly interacting with each other, and those interactions are important. So when you look at this particular environment, you can see all the different animals. So they are interacting with their environment. You can see that the deer depend on the grasses, right? Because they're going to use that as a food source. The fish is dependent on being in a water environment. It's also dependent on whatever its food source is, insects or smaller fish. The plants can absorb absorb carbon dioxide from the air and then release oxygen so that the animals can breathe that. Then the animals take in the oxygen and eliminate the waste, the carbon dioxide, which the plants can then use. So again, these organisms are all interacting with each other and with the environment, and it's an important part of stability of this ecosystem. This is just a quote. It says, in biology, nothing makes sense except in the light of evolution. So we'll kind of talk about that a little bit. Evolution, unity, and diversity. Diversity and unity are the dual faces of life on Earth. So they kind of mean different things, right? Diversity means differences, and unity means like coming together, similarities. So 2 different things. So it's kind of hard to imagine that they're so related, but in fact they really are. Scientists estimate that there are probably somewhere between 10 and 200 million different species out there. This is really hard to pinpoint down. That this is a huge range, I know, and that's because it's very difficult to count microscopic organisms or highly mobile organisms or organisms that hide, like insects that can get in really, really small places, or the oceans are deep and hard to explore, very expensive to explore, and so again, there's a lot we don't know, and we don't actually know how much we don't know. They have, however, identified and named about 1.8 million species. Taxonomy is a branch of biology that helps us name and classify organisms. That's really important to do because that's a lot of data if you consider 1.8 million species, and we need a way to manage that data. Taxonomy allows for us to categorize all these differences, all this diversity, which makes it more manageable. It allows us to be able to understand which organisms are more closely related to each other and which organisms are more distantly related to other organisms, right? So the closer they are on a branch together, the more they share in common, the more related they are. The farther the branches are from other branches, the less they have in common. The more distant the common ancestor would have been. There are 3 domains of life, and so this is the first level of taxa, the first level of taxonomy. So again, 3 domains. 2 of these domains are prokaryotic domains, so single-celled organisms. So these are essentially 2 bacterial domains almost. Prokaryotes were so diversely different that they had to be divided into 2 different domains. These 2 domains used to be categorized into 1 domain, but that had to be changed again because these 2 groups were very different from each other. So one is called domain bacteria and the other is called domain Archaea. Prokaryotes are single-celled organisms that lack a nucleus, and so that's true of both of these domains of organisms. The third domain is domain eukarya or domain eukaryota, same thing. And this includes all eukaryotes. So I always tell students, you are a eukaryote, and then hopefully that will help you remember that you belong to this category. Being a eukaryote typically means that you have a nucleus. Each domain is then subdivided into kingdoms. There's a lot of different kingdoms. This is going to show you only the kingdoms that fall, like the major kingdoms that fall under eukaryote domain. We don't really further classify in this class or even in IB beyond just domain bacteria or Archaea. But we do under eukaryotic domain. So we have kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. So Animalia is all animals, Plantae is all plants, fungi are all fungi, and then everything else that is a eukaryote that's not a fungus, not a plant, or not an animal kind of gets put into this catch-all category of protists. They tend to be unicellular, but some are multicellular, and they are just more complex compared to the prokaryotic domains. Beyond kingdom, we will further classify as well. So here are the letters of the different levels of classification, and then here are the words. So domain, remember, is the first, so we have domain first, and then K stands for kingdom, P stands for phylum, C stands for class, O for order, F for family, G for genus, S for species. So kingdom, or it's domain first, and then kingdom, phylum, class, order, family, genus, species. So you do need to know the order. Domain is the largest, most broad category or taxa, and then species is the most exclusive category. It's the smallest category. Members of the same biological species can breed with one another to make fertile offspring, whereas that wouldn't be true if they only shared a genus or they only shared the same family. But if they share the same species, then they also share all these other categories above it. That's how this works. But it's possible they could be in the same domain and not be in the same kingdom, or be in the same kingdom but belong to different phyla, right? So it only kind of works one way and not the other way. This is a link to a classification rap that I'm going to play. I am hoping that you'll be able to hear the sound through this recording device. So let's see. Okay, so before we watch the song, it's like an 80s rap, but it like really gets stuck, probably 90s actually, really gets stuck in your head though, and really helps students. Like you'll probably never forget it. The order of kingdom, phylum, class, over order, family, genus, species. Kids come in the day of the finals singing it. I mean, they come into IB 2 years later singing this song. It's just kind of catchy. So here we go. So that song is linked to the PowerPoint if you decide you'd like to see it again. Okay, so again, moving on with this same theme, unity in the diversity of life. We know that life is unified because there is a universal genetic code. So it's the same code, the same 4 letters, the same 4 bases in DNA for any organism. So bacteria have the same code as you do, as plants do. In fact, we can read each other's codes. We can cut out a piece of human DNA, insert it into bacteria, and they can read our code and make our proteins. Big industries like Amgen do this all the time to help put out pharmaceutical products like insulin and things like that. Similar metabolic pathways. So all organisms essentially perform cellular respiration. We have to have ways to break down the foods that we either take in or make if they photosynthesize and convert them into smaller packets of usable chemical energy called ATP. So all living organisms do that, but how they accomplish it is a little different. And then similarities in cell structure. There are certain structures that all cells have. All cells have a cell membrane. All cells have cytoplasm. All cells have ribosomes, the organelle that's used to make proteins. Right? These are things that all cells have in common. Evolution helps us to understand the similarities that are present, even though life on Earth is incredibly diverse. Evolution is the core underlying theme of biology. So it really kind of helps bring everything together and make sort of everything that we learn throughout a biology class make sense. It's kind of one of the reasons why we do the evolution unit last this year, because by the time we get there, you'll have so much information, and that unit will really help you kind of put all the different pieces of the puzzle together. So here's an example down here, which shows a group of bacteria, and then it shows that we've put antibiotics on this group of bacteria, and most of them died. So all these gray ones die. Well, what you have left with are bacteria that are resistant to the antibiotics, and all the ones that are not resistant die. All the ones that are resistant live. So typically antibiotics kill the bacteria that causes you to be sick, but they can also kill good bacteria that your body needs to help protect you. And then what happens is, well, now you have these resistant bacteria, and then the resistant bacteria divide to make more resistant bacteria. They don't have any more competition from the other non-resistant bacteria, so they quickly outnumber those. And then another thing that bacteria can actually do is transfer resistance, so transfer genetic information from one bacteria to another, which can just further complicate this process. So life evolves, changes over time. Okay, moving on to the scientific process. You guys have probably had the scientific process every year since the 6th grade. So this isn't really going to be anything different. One thing we really want you to understand, though, is that the scientific method does not have to be rigid. It's not linear. You don't have to start at one side of the scientific method, follow every step until you get to the end of the scientific method. There's all this looping involved, right? Like you can go back and rethink things, redesign your experiment, redo the experiment, collect data a different way, and so on. So again, doesn't have to be rigid or fixed. It's not linear. It's sort of like evolving, flowing. Scientific inquiry and investigation do involve what we call hypothetico-deductive reasoning. And so what that means is that you have like a prediction or like a question that you're asking. So if this is happening, then this is what I think will happen because of this. It doesn't have to be in an if-then statement, but it kind of helps to set it up, your hypothesis, as an if-then statement. So if eating chocolate affects acne, then those students who ate the chocolate will have more acne than those who did not. Science involves different variables. We have our independent variable. This is what you are actually changing as the experimenter in order to test your hypothesis. The independent variable is sometimes called the manipulated variable. So I would write that down. They're the same thing. The independent variable is the same thing as a manipulated variable, because you might hear that. The dependent variable is, what are you measuring? What are you actually collecting as your data? And this data collection is thought to have changed because of your independent variable. Right, so the dependent variable changes based on the independent variable. It's sometimes called the responding variable. I would write that down. The dependent and the responding variable are the same thing. And then controlled variables are everything that you're holding constant in your experiment. It's really important that you only have one independent variable and that you don't have more than one. Every other variable should be controlled. Because if you don't control them, how do you know what's causing the change? How do you know that your independent variable is actually affecting your dependent variable? You wouldn't, because you're changing too many things. So one independent variable, but then keep everything else constant. Those are controlled variables. Sometimes you'll have what's called a controlled group. That's different than a controlled variable. Not every experiment will have this, but if you have a control group, you also have an experimental group. And the experimental group is going to receive whatever variable that you're trying to test, whereas the control group will not get that. And so that way you'll know if a change is happening, it's likely due to whatever variable you're giving the experimental group. So here we have a plant pot that's getting water. There's seeds in there. And then this control group gets no water. So after time, this pot has sprouted and this pot has not. The control group did not sprout any plants. Therefore, water must be affecting seed sprouting. There's two types of data, qualitative and quantitative. Qualitative data involves descriptions, so using your senses to collect data, but quantitative data must involve numbers, so N for number. I always tell students to underline the N. Observation versus inference. An observation is something that you can use your senses for to actually like see or smell or taste. It can be qualitative or quantitative, but you have to be able to actually use your senses to gather that information, right? So we could obviously see that there is a ring on this finger. We can count the number of fingers this person has, things like that. Those are observations. But an inference is a conclusion that you draw based on your observation. So when I look at this hand, I might think that this person is engaged or married because of the ring on the ring finger. And I might not be right. My inference does not have to be right. There could be a lot of reasons why this person is wearing the ring on this finger, but your inference does have to be logical, right? There has to be a reason why you think that. So you can't just like make something up. It does have to make sense. And then this shows you that, you know, something is happening here. You can see there's like a rift here, and you can see that damage has been done, right? So those are all observations. I could also quantify things. I could count how many trees, count how many houses, things like that. But then I can infer that some type of disaster has happened, so maybe an earthquake. Be careful when you're collecting data that you're only putting observations in the data table. You're not making any inferences. That would be saved for like a conclusion section of the lab report. Okay, we're going to talk about proper data table formatting. And this formatting is aligned with IB. So it's really important that you do always come back to your notes when you're forming data tables, because your data tables will always be set up the exact same way. So you must have a title, and your data table must have a number, even if there's only one data table in your whole experiment, data table 1, and then a highly descriptive title after that. This title needs to include, at minimum, the dependent variable and the independent variable, but it also should include anything else that's needed in the title. If you use an organism, if you use special equipment to collect the data, like anything you would need to know as a reader to understand what your data table was about, that needs to be in the title. So I would rather you guys give me long descriptive titles in the beginning. It's a lot easier for me to like kind of tell you what you don't need than it is to tell you what you're lacking. Okay, so detailed titles. So that's kind of gone over here, right? Table number, descriptive title, the variables, the length of time. If you used a living organism, you would use the scientific name, so genus and species, for example. And then when you set up your data table, notice that all my headers are up here at the top of the data table. This is the information that describes the actual data that I'm going to put inside my table. So you're never going to put these words down the side. Like, that's not the proper way. That's not the way IB sets up their data tables. All these headers go at the top, and then it will be understood that the numbers you record or information you record below it belongs to this category. Typically, the independent variable is on the left side of the table, followed by the dependent variable, and then followed by anything else that the experiment might have you do. And there can be more columns. So if you had to record a calculation, like an average, that could go in this column. If you also needed to record observations, then there could be a fourth column. Kind of just depends on your lab and what's required. The units only go up here in the header. The units do not need to go down here in the actual data table. That's considered redundant. Again, it's understood that whatever falls below here belongs to this header. This header is describing this information. The table is read vertically. And then anytime you do a calculation, it goes underneath the data table. You need to have a sample calculation. You'll never do math inside the table. Sample calcs underneath, and then just the final answer here with the unit up here. Please don't write IDV for independent variable. You'll replace it with the independent variable. So if you were doing an experiment with plants where you were giving different pots of plants different amounts of water, then my independent variable is the amounts of water. And so I wouldn't write IDV here. I would write amount of water. And then I would have to include a unit with this as well, so milliliters. And then, again, I would replace these words with the actual dependent variable of the experiment. So plant height, and then in centimeters. If I used a ruler to measure, I'd have precision that I'd add here. So plus or minus 0.5 millimeters or something like that. And then again, same thing for this column. So these are just examples of tables, so you can kind of see how tables are actually set up, right? So I have my title. Of course, you need a number, data table 1, and then a highly descriptive title that goes above the table. And then below or in here, I have my actual data table. Here's my headers. Notice how neat and tidy it is. Since we are remote this year, then it will be actually really good for you guys to practice tables electronically. Like, you can set up tables in Google Docs, you can set it up in Word, you can set them up in Microsoft Excel, whatever you're comfortable with, and you can like merge different cells together. That's how I have this one big cell over top of this cell, right? That's how I have this cell taking up the same amount of space as two cells are here. And I can show you guys how to do this stuff if you don't know how to do it. But basically, here's my independent variable on the left. Here's my dependent variable with a unit plus or minus the precision of the instrument. And then here, and that's not always needed, here I have separated my dependent variable into 3 columns because I did 3 different trials, right? So the data for trial 1 goes here, trial 2 goes here, and trial 3 goes here. This would be different levels of independent variable. That's why there's multiple rows here. So for example, if we go back to the plant getting watered with different amounts of water experiment, then this would be amount of water in milliliters, and I would have 3 levels here. So the first plant's getting 10 milliliters, the next one's getting 20, and the next set of plants are getting 30 milliliters. And then if I wanted to calculate an average of all 3 trials, then that average would go here. And then here's just more data tables that you guys can look at. Again, they're all formulated the same way, so please really pay attention to that because you could be scored down if you're not formatting tables the same. Just refer back to your notes and ask questions. Graphs also have very specific formats. They need a figure number and they need a title. If you're just taking data from a data table and graphing it, then you can use the same title. It's just that you'll have to write figure and then a number, and then just copy and paste your title right here. Figure titles go under graphs, though, not on top of graphs. That's different than a data table. The data table title goes on top of the data table. when you actually graph the data, you'll have two axes, the y-axis and the x-axis. The y is the vertical axis, the x is the horizontal axis. Your independent variable always goes down here on the x, and your dependent variable always goes on the y-axis. This is really good to help you remember. Dry mix. So dry mix. And what this stands for is the dependent or responding variable on the y-axis, the manipulated or independent variable on the x-axis. So if you can remember dry mix, that will help you remember what goes on what axis. And then if you have to have a scale because you have numbers, make sure it's at regular intervals. So it can't be like 1 and then you go to 5 and then you go to 7 and then you go to 15. It has to be like count by 5s or count by 1s, something like that. If you have more than one dependent variable, which we don't do very often in this class, then you would need a legend. So that's not more than one level of independent variable. That's not the same thing. More than one dependent variable, you would need a legend. If you don't actually need a legend, then please don't put one. Units should go with the labels only and not with anything else. Okay, so that's actually shown here. So amount of fertilizer, and here's my units. It's in grams. So notice that I didn't write 10 grams, 20 grams, 30 grams. That's redundant. I wrote the unit here, and it's understood that this means 10 grams. The same for here, right? Plant height in meters. So 0.5, 1, 1.5. No units go on the actual axis with the number. They go over here with the label. And then my figure number with a highly descriptive title under the data table, not above it. So just refer back to your notes to help you. This is just a flowchart to help you kind of figure out, well, what kind of graph do I do? So you can try following this key. Is the data a percent that sums up to 100% or some kind of a total? Like, is it a proportion of a whole? If yes, it could be a pie chart. So that's an example here. If it's not, you wouldn't use a pie chart. So you can go over here. Are both manipulated and responding variables quantitative? Are they numbers? If they're not numbers, like one is a description, like let's say instead of changing the amount of water, I was just doing descriptive temperatures, not actual temperature, but more like cold water, room water, and hot water, or cold, warm, hot. Those descriptions means that you're using words, and I didn't actually put a thermometer in. It was more subjective. And so then I would use a bar graph. If you are using quantitative data, numbers for both, then you'll choose a different graph. So go here. Is your manipulated variables continuous? Like, does it follow along an interval? Is it time? Could it be a decimal? Something like that. Continuous data. Or are you clumping it into specific groups? So if it's continuous, then it would be a scatter plot or a line graph. Lines specifically mean a change over time, whereas a scatter plot is specifically showing a relationship with the two variables between x and y. Be careful with line graphs. It doesn't always mean you need to connect the dots. Sometimes it's a lot better to show a trend line that goes through the data rather than an actual line. So really think about what that means. A line would mean, if you're connecting the dots, that like each data point is connected to the next continuously over that interval. If that's not true, then just draw a trend line, even if your trend line is a curve like this. If it's clumped, the data is clumped into groups like 0 to 5 years, 6 to 10 years, then you could use a histogram. A histogram is essentially a bar graph, but with numbers on both axes. You will need to be able to convert scientific units in this class. So we're just going to do metric to metric conversions. We won't do English to metric. So you'll always use the metric scale since this is a science class, and you'll never use something like inches. It's always centimeters and millimeters, things like that. We'll never use inches. And in this class, again, we won't convert like inches to centimeters, just centimeters to millimeters to kilometers, things like that. And I'm going to show you how to do this. So here's the most common prefixes that go before whatever unit you're measuring. So let's say we were using grams as our basic unit. It could be kilograms, hectograms, decagrams, grams, decigrams, centigrams, milligrams. And so you're going to need to remember the order of these units. And there's little like sayings to help you do that. Here's one. King Henry doesn't usually drink chocolate milk. So king stands for kilo, Henry for hecto, doesn't for deca, usually is for the basic unit. So that could be grams if you're measuring mass. It could be meters if you're measuring length. It could be liters if you're measuring volume. Drink, deci, chocolate, centi, milk. King Henry doesn't usually drink chocolate milk. Another one I really like is kind humans don't make dogs climb mountains. So kind humans don't make. Make stands for the basic unit, which in that case we're talking about meters, but any unit would work. So again, kind humans don't make dogs climb mountains. Same thing. So whatever helps you remember, you do need to remember the order. In bio, we'll also use this very, very tiny unit that's called the micro whatever. So microgram, micrometer, microliter. There's 1,000 micrometers, for example, in a millimeter. So it's very, very tiny. So whenever you're using this metric conversion, we're talking about moving the decimal over. Okay, so if we're going from kilo to deca, for example, then I'm moving my decimal over 2 places, and I just move to the right, right? So I'm just moving it over to the right 2 places. That's the same thing as saying multiply by 10, multiply by 10 again. So notice the blue arrow goes this way. That means every time you move a step on the metric ladder, then you would have to multiply by 10 if you're moving from left to right, or just move the decimal over that many spots. If you're moving from right to left, then you'll divide by 10 each time, or move the decimal to the left, however many times you move. So if I'm going from milli to centi, then I move my decimal to the left 1 time, or I divide by 10 1 time. There's some examples on the next page. Okay, so I want to change 2.3 kilometers to meters. So I have to start here at kilo, and I need to count how many spaces I'm moving it over. So kind humans don't make dogs climb mountains. So my basic unit is here. Meters is a basic unit. So 1, 2, 3. So I need to move my decimal place over 3 spaces. So 1, and I needed to add zeros to see this, 2, 3. So 2.3 kilometers equals 2,300 meters. So you guys will need to practice these. And then here's micro down here. Micro, you have to move it 3 spaces down from milli. Okay, so if I wanted to convert, let's say I wanted to convert, let's just say 3 millimeters, right, to micrometers. So I have to move it down. This is 3 spaces because it's not just times 10, right? It's times 10 3 times. So if it's 3 millimeters, then I have to move the decimal over 3. 1, 2, 3, be 3,000 micrometers. And the last chart showed you that. So let me go back and show you that. Right here. So instead of being 1 space, 1 decimal place between these two, there's 3. So please try and remember that. Micro is written like a little kind of cursive U, but it's micro. Okay, and that is the end of the Chapter 1 notes, guys. Good job.